Thank you, Linek, for the introduction, and good afternoon, everyone. Now I will present you part of the BIODHYL project based on in situ surveys that aim to measure the biofouling variability depending on sites, depths, and time. So this part of the project aimed at reaching different objectives. The first one was to design a system for in situ monitoring ahead of the installation of floating offshore wind farms and enabling the sampling of biofouling at different depths. The second objective was to deploy these systems offshore Atlantic and the Mediterranean Sea. Then to measure biofouling in terms of weight, volume, and biodiversity across different spatial and temporal scales, and also considering different materials representative of dynamic cables and synthetic mooring lines of floating offshore wind farms. And to measure also environmental variables such as temperature, salinity, nutrients, pH, etc., along with the biocolonisation survey. And the overall objective was to gather quantitative and qualitative data to characterize the biofouling at offshore sites based on direct measurements, so the measurements that are done directly after retrieving the samples, so the weight measures, the volume measures, the thickness, etc., and based also on morphological analysis and molecular analysis that have been conducted in the lab after the sampling. But for this presentation today, I will focus on weight and biodiversity data across different sites and at different times, based on the direct measurements and only on morphological analysis. So for these surveys, we have deployed our systems at 2 sites in the Atlantic and at 4 sites in the Mediterranean Sea. So you see the 3 letters for each site is the name of the buoys, under which for the fine time and space monitoring, we deployed systems and samples at 3 depths under 3 buoys, so MemoFlow and Omega M and Ome in the Mediterranean, and Apil, which name is GBI, in the Atlantic. And for long-term monitoring, we deployed plates, PVC plates, under 3 buoys at subsurface. So offshore Leucate and Cap Couronne in the Mediterranean, so the Bob and Cap buoys, and under Basse des Chats buoy in the Atlantic, the BDC. And these deployments were not possible without the help and the collaboration of with Farz et Balise, Écocéan, the CEFREM Lab also, and the Parc Marin de la Côte Bleue, which were not directly involved as partners in the BIODHYL project, and I wanted to thank for their help for these deployments. And the systems we deployed are like this. So you see here some the designs of the systems that we've been doing at France Énergie Marine, and some views also of the systems during deployment and just after the deployment. So undersea views as well of the systems, with part of them are biocolonized already, and 2 views also on different sensors that we deployed here for measuring acceleration of the lines, the mooring lines, and also multi-parameter probe. So concretely, after retrieving the samples, we measured the weight of the biocolonization in air and in water using dynamometers. We also measured the thickness of the biofouling, so through the measurement of the length of organisms and also measuring the perimeters of the samples biocolonized. We also measured volume of the biofouling precisely through water displacement, but we also calculate the volume based on thickness measurements. And later in the lab, after sorting the samples, species richness has been measured, as well as the abundance of the organisms for each samples and the coverage also of the organisms of each samples. And as I said, we used also sampling and probes to measure different physical, chemical parameters. So we gathered lots of environmental data, as well as quite a lot of biofouling measurements. So for example, we measured more than 100 volumes, almost 2,000 weights, and over 1,000 biofouling thicknesses. And we also taken pictures, including pictures with known distance during the surveys. And regarding the taxonomic analysis, so we identified 246 species during the BIODHYL project, so based only on the BIODHYL samples. And we also identified a lot of species based on molecular analysis, but this will be presented later. And today, so I will present a selection of some key results, starting with the biofouling variability in terms of weight along time. So in these figures, you see the wet weight measured in air in kilogram per square meters along the biofouling age, which is there expressed as years, and at the different sites at which we sampled the biofouling. So you see that there is a global increase in wet weights along time. For example, for the samples at GBI, in the Atlantic, there is an increase from 6 months to 2 years old samples, and there are also some differences across sites. Then looking at the variation of biofouling weights along depths, so you see here the weight in air and the depths there, and these data were collected after only 1 year of immersion. So at these two sites, MEM in the Mediterranean and GBI in the Atlantic. And you can see that there is a global decrease of the weights, of the biofouling weight, with increasing depths. And that the weight variation tended to decrease along depth, which is an interesting note. But also more data are needed to confirm this trend. Then we wanted to present you data regarding the density of biofouling expressed as rho, so which is the density in kilogram per cubic meters, calculated based on the wet weight measured in water, as Guillaume has presented you earlier. And here you can see the mean density, box plot of the density in the Mediterranean and the Atlantic according to different depths. So in blue, low depths, in red, medium depths, and in green, bigger depths. And we can see that the biofouling density is globally lower than the value provided by standards. So the red line on the graph, which is the standard biofouling density of 130 and 25 kilogram per cubic meters. So this is true for almost all the data except the biofouling that was measured at the Atlantic site at 40-meter depths. And there the biofouling amount was low, so there is maybe the sensitivity of the measure which affect also these results. And also, there is an increase, a tendency to increase in density with the water depths. And overall, the mean biofouling density was 215 kilograms per cubic meters, which is lower than the standard. Then looking at some diversity indices across time, we wanted to show you the species richness, the mean species richness at the different sites surveyed a long time. So you see there that the mean species richness increased a long time, with a notable decrease at 2 years for these 3 sites, so Cap and Bob in the Mediterranean, as well as Basse des Chats in the Atlantic, which is actually linked to a step in the succession. We'll see that a little bit later. And the mean species richness varied between 8 at the beginning of the biocolonization and 45 later in the process. And regarding the variability of the species richness along depth, we see that on this graph there is no really clear patterns regarding this variability along depth, and the patterns observed vary depending on the age of biofouling, so either 6 months or half a year, 1 year, 1.5 years, and 2 years of the biofouling. So we don't see the same patterns for this. Then regarding the community structures of the biofouling observed at one site, we have other observations at the other sites, but I wanted to present you these figures today. So you can see here a multivariate analysis, an ACP, which shows the different samples collected at the Basse des Chats. So each dot is one sample, and when the dots are close together, it means that the community structure, the assemblage of the organisms, are more similar. So we can see that across time there is a change in community structure. So with the community at 6 months or age of 6 months on this part of the analysis, then after 1 year on this part of the analysis in orange, and after 4 years they are in blue. And different species actually drives the structure of the community. So the small amphipod Jassa for the youngest samples, then Mytilus for the community after 1 year, and small species living within the mussels after 4 years. So just for the pleasure, some pictures of the different organisms that we encountered in the field and in the lab. And as take-home messages, so this project, the BIODHYL project, enables the design of sampling devices that are adapted to dynamic offshore environments and which enable the samples of biofouling at different depths. Also, as take-home messages, our surveys underline that biofouling wet mass increase over time but decrease along depths, and that the biofouling density tended to decrease with depths and varied across sites. Also, regarding biodiversity, biofouling species richness increase over time, while the community structures change over time also, with the general succession, the small amphipod Jassa, then Mytilus, and then the species associated with Mytilus, which grow on the mussels or that lives around between the mussels. And these projects enabled us also to gain robust quantitative data on 2 facets of interest for offshore wind, the Atlantic and the Mediterranean, at different depths and at different times. And the development of BIODHYL devices is relevant for environmental characterization phases, to refine the structure design to specific sites, and also to measure parts of the reef effect. And as perspective, we think that using this approach more offshore and for example, for the Call for Thunder 10 in France, and also at larger spatial scale, for example, at the scale of the Mediterranean Sea, to foresee the biofueling potential at sites where the floating offshore wind farms will be developed would be useful. Then there is a need for longer-term surveys in order to identify the succession steps and also the effect of climate change. And there is a need to gain data on biofueling communities within offshore wind farms by developing adapted methodological approaches. So after gaining knowledge before the installation, then we need to gather knowledge within service offshore wind farms. And also to develop integrating tool to show variability and uncertainty of biofueling for design. And finally, probably extend the biodiversity surveys to biocolonization, which include mobile species, not only the fixed species. I wanted to acknowledge many people and again underline the founders of this project, which enable us to conduct these surveys. And thank you for your attention.